Okay, hi everyone. So this is a recording of me discussing the lecture slides from lecture one that I didn't quite make it to. What I want to do is just talk through these slides just as I would normally in a lecture. Just explain what they mean, talk around them. I know some students will look at the slides kind of by themselves, and that's absolutely fine. There is extra information though, there's exposition. There's real value in having me explain these slides to you because I've composed them. I understand them really well, and there's extra bits of useful information I can give. Okay, so firstly, I wanted to talk through the microorganisms. So the first portion of the module is called the microorganisms, and this is the portion of the module in which we talk through the different groups of microorganism one by one, and we essentially understand what each group is, why it's a group, what defines that group. And it's super important because the second part of the module is infectious disease. And in this part of the module, we're talking about how these different microorganisms cause disease, the way they cause disease, what types of diseases they cause. And crucially, we're talking about how to diagnose this disease and how to treat this disease and how to prevent this disease and all these different things that are so important to human health and so important in medical science. To understand those elements, we need to understand the microorganisms themselves. So if you want to understand how bacteria cause disease and how we can prevent bacterial disease and diagnose it, we need to understand what bacteria are. If we're treating bacteria with antibiotics and we're using a specific type of antibiotic and we're saying that antibiotic has something to do with peptidoglycan, we need to understand what peptidoglycan is. If we're using an antibiotic that inhibits the method of replication used by a bacteria, we need to understand what that method of replication is. Same for viruses, same for protozoa, same for fungi, and same for the parasitic worms. So that's the purpose of the first part of the module, and that's kind of what I'm going to introduce today. I'm going to go through these groups and we're going to look at the key bits of information we need. So first off, we're going to talk about bacteria. It's perhaps the archetypal group of microorganism. When we think of microorganisms, our brain usually goes straight to bacteria, straight to germs. So they're fairly well known, but we need to understand them in a little bit more detail than perhaps the understanding we have in a general basic level. We need to understand exactly what they're composed of. And to understand that, we can compare a bacterial cell to a eukaryotic cell. And that's what we have here. The bacterial cell is actually labeled as a prokaryote. The reason for that is because bacteria are prokaryotes. So cellular life can be eukaryotic or prokaryotic. And there are fundamental differences between eukaryotic cells and prokaryotic cells. Our cells are prokaryotic, bacterial cells, our cells are eukaryotic, sorry, bacterial cells are prokaryotic. So our cells have the features described on the bottom in this diagram. They have a nucleus, they have lysosomes, they have endoplasmic reticulum, they have mitochondria. Bacterial cells don't. They don't have any of these features. They're prokaryotic, and prokaryotes don't have these features. Prokaryotes have a nucleoid instead of a nucleus, so they don't have this distinct membrane-bound nucleus. They have a nucleoid, kind of a sprawling jumble of DNA. Their genome works the same way as ours by and large. It's nucleic acid. It encodes the RNAs and that will eventually be translated into proteins. So the principles are all the same. It's just it's a nucleolus rather than a nucleus. That's the term we use to describe where the genome is. Bacteria also have what we call plasmids, and these are really important. They're circular portions of the DNA. It's difficult whether we refer to them as the genome. Technically, they are the genome because they're part of the bacterial cells' genetic information, but they're separate from the main genome. They're separate from the nucleolus. So it's important we understand they exist. We're going to talk about why a bit later in this lecture and in lecture 2 as well. There's some other features here that are perhaps not super interesting, but the one that is interesting and that we're going to talk about at length is the cell wall. So the cell wall surrounds the bacterial cell. It gives it structure, it gives it protection, really important feature. Different bacteria have different types of cell wall, and we're going to talk about what the implications of the different cell wall types are. It's important to understand as well that even though all bacteria are prokaryotes, not all prokaryotes are bacteria. So there's this separate group of life. They're unicellular, they're composed of single cells, just like bacteria, because all prokaryotes are single-celled, whereas a lot of eukaryotes, like us, are multicellular. But this other group of prokaryotes, they're not bacteria, they're archaea. They're very different from bacteria in a lot of ways. Basically, the only thing they have in common is that they're both prokaryotes. We are going to talk about archaea a little bit in later lectures, but they're not particularly interesting in the context of this module. We're just going to talk about them from an evolutionary perspective, because we're going to look at the tree of life and look how the different microorganisms evolved from each other. And when we're doing that, it's interesting to see where archaea are, because essentially we have 3 domains. We can split all of life into 3 branches. One are the eukaryotes, including us, another the bacteria, and another the archaea. So archaea exist as their own separate branch, and both the archaea and the bacteria are prokaryotic branches. So there's this idea of gram-negative and gram-positive. And when I said the bacterial cell wall can have different kind of compositions, this is what I was referring to. The bacterial cell wall can be gram-positive or gram-negative. Gram-positive bacterial cell walls have a thick layer of peptidoglycan, whereas gram-negative bacterial cell walls don't. They have a thin layer of peptidoglycan, and then they have a layer of LPS on the outside. That probably doesn't mean much to you right now, but when we talk through specifically what the implications of this peptidoglycan layer are, or what the implications of the LPS layer are, you'll understand just how important it is to understand whether or not bacteria are gram-positive or gram-negative. It has huge implications for the bacteria. You may have heard of gram-positive and gram-negative bacteria. You may have also heard of something called gram staining. Gram staining is how we tell the difference between gram-positive and gram-negative bacteria. If you haven't, don't worry, because we're going to cover it in quite a bit of depth. Again, it's really, really important. There's some other bits of information you need to understand about bacteria here. As well as classifying bacteria as gram-negative or gram-positive, we can classify them based on their shape. So this isn't to do with the composition of the cell wall. This is to do with the overall shape of the bacteria, the morphology. So some can be cocci or coccus, basically circles. If you look at them through the microscope, they look like tiny little spheres. That's their morphology. Some can be bacilli, and these are essentially elongated rods. Again, if you look at them through the microscope, you just see elongated rods. They look like capsules, sort of like capsules shaped like a tablet. If you Google this, you'll see plenty of different images. If you look at the textbooks that we have on the course, you'll see a lot of images as well. Very clear to distinct, very clear and very distinct. Spirochetes are this kind of squiggly shape. So it's kind of it's almost like a swirl, but it doesn't always look quite like a swirl. It just looks somewhat irregular. Doesn't always look quite as neat as it does in this graphic. This graphic is from MIM's Medical Microbiology, by the way, which is a really good book. It's a little bit advanced for level 4, but if you are finding this stuff interesting and you are finding you want to push forward, I would encourage you to take a look at it. We can also classify bacteria in other ways, such as how do they grow, what environments do they grow in, do they need oxygen or do they not need oxygen? Again, we'll look at that specifically in the bacteria lecture, but for now, I'm just introducing the idea that we can understand bacteria in this way. We can say, does this bacteria need oxygen to grow? Does this bacteria get its carbon from CO2, or does it get it from glucose? These kind of technical questions that when we start to answer them, we start to understand how different bacteria are, and we start to put them into different groups. Okay, here's an example. So I just kind of mentioned that different bacteria will have different requirements in terms of oxygen. So we can consider some bacteria obligate aerobes, so they need oxygen. We have microaerophiles, so they can grow in small amounts of oxygen. We have obligate anaerobes, so anaerobic, no oxygen. So these will grow in the, these won't grow if oxygen is present. Then we have facultative anaerobes or facultative aerobes, and they'll grow under both conditions. They can kind of switch between the two, dependent upon the conditions. Don't need to know any more details about that now. Just need to understand the idea that we can classify these bacteria based upon their growth conditions. This is a really important graph. So it's basically it's not really a curve, but we call it a growth curve for bacteria. And what you'll notice is this logarithmic phase here, where it's going straight up. This is what we call exponential growth. The reason why we see exponential growth in bacteria is because they can reproduce by something called binary fission. Essentially, it means splitting in half. Splitting in half, then they grow, they make more proteins, blah, 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 blah. Then they split in half again, go through the process, split in half again. So they're doubling every X amount of time, however long it takes them to undergo each cycle. And again, that differs by bacterial species. They're doubling. Kind of scary, actually, because you think, dependent upon how long that cycle takes, you're looking at billions and billions of bacteria being produced. And beyond that, if you get continued exponential growth, trillions, and exponential growth is kind of a scary thing, because the longer it goes on, the bigger these doubles become. And you can potentially go from just a small number of bacteria, or even one, to huge numbers very quickly. The good news is that even though there are a lot of bacteria, there's not kind of meters thick packed bacteria or anything. There's not exponential growth going on for most bacteria under normal conditions. The reason is that there's this stationary phase. So bacteria will divide and divide and divide when they can. So under ideal conditions, if they have the nutrients, if they're in the right conditions to do that, then they're able to do that. However, they're not always in the right conditions. There are limitations. Each particular environment will run out of food. There won't be the particular oxygen requirements. There'll be competition from other bacterial species. There'll be competition within the bacterial species. So this exponential growth, whilst it does happen, it's not indefinite. So whenever you grow bacteria, usually what you'll see is this logarithmic phase, the exponential phase, then a stationary phase, so where this exponential growth stops, and then often you'll see this death phase as well, and we'll go into details about that. But it's to do with the number of bacteria that can be sustained in a particular environment. Okay, so very briefly, I want to introduce some of the bacterial diseases that we'll be talking about in the infectious disease portion of the module. You'll probably know some of these. You might not know that they're bacterial diseases. It's quite easy to confuse bacterial diseases with viral diseases and fungal diseases even, because a lot of the symptoms are the same. For example, if you have a bacterial infection of the GI tract, which in this example on this slide is by cholera, you'll almost invariably get quite severe diarrhea. There's a lot of viral infections of the lower GI tract, the enteric viruses, that also cause diarrhea. So quite similar symptoms, but the way these symptoms are caused is quite different. That concept, the way the microorganism causes disease, is called pathogenesis. Genesis meaning how something originates, patho meaning sickness, suffering, illness, like pathology is the study of sickness and illness, psychopath, psychomind, path, ill, dysfunction, that kind of thing. So we're looking at the origin of illness, the origin of suffering and dysfunction, pathogenesis, in the context of these microorganisms. For cholera specifically, the causative agent, so the bacteria that's causing it, is Vibrio cholera. And what Vibrio cholera does is produces a toxin, so produces a protein, and this protein induces severe diarrhea. This isn't unusual. A lot of bacteria produce proteins. Again, we usually call them toxins, specifically exotoxins. So an exotoxin is a toxin that the bacteria releases. Cholera is a good example. That's why I've put it here. It's the main example. It's the clearest example, if you like. But yeah, I just wanted to introduce the concept. It's treated with antibiotics. Again, we should know that bacterial infections are usually treated with antibiotics. Antibiotics are never used against viruses or anything like that because they don't work. Antibiotics specifically target bacterial processes and bacterial structures. So we want to get that really clear straight away. Antibiotics for bacteria. For virals, we use antivirals. For fungi, we use antifungals. We don't want to slip into this confusion of using antibiotics for viral infections. It's just like kind of this fundamental principle that we should establish now. E. coli gastroenteritis, very similar. You get E. coli colonization of the lower GI tract. By GI tract, I'm talking about the gastrointestinal tract. So mouth, throat, stomach, intestines. When we're saying lower GI tract, we're talking about the intestines. A lot of microorganisms that cause diarrhea will be colonizing the lower GI tract. Again, Vibrio cholera is an example, and we have E. coli as an example as well. You'll often use E. coli in the lab. The E. coli you're using is what we call non-pathogenic. So again, there's that term pathogenic, the origin of disease. Non-pathogenic E. coli is not capable of causing disease because it's been weakened. Certain genes have been taken out. So the type of E. coli we use in the lab, at university laboratories at least, is non-pathogenic. It's been deliberately weakened to make it safe. Some other bacterial infections. So I wanted to mention streptococcal pharyngitis, which is strep throat essentially. It's a streptococcal infection of the respiratory tract, of the pharynx really. So when we say itis means inflammation, and inflammation is a response by the immune system to infection. So if we say meningitis, meningitis means inflammation and infection of the meninges. Laryngitis, inflammation, infection of the larynx. Pharyngitis, inflammation and infection of the pharynges, of the pharynx. And that's what we see in pharyngitis, and that's what we see in streptococcal pharyngitis. We have Streptococcus pyogenes, just a specific species of bacteria infecting the pharynx, and then an individual has inflammation there, which is a localized immune response. And this localized immune response is experienced as a sore throat That's really what a sore throat is in infection. It's your immune system detecting a pathogen there and launching an assault to try and get rid of it, but actually kind of potentially doing more harm than good, at least in terms of your own comfort, because sore throat's not very nice, but it is a strategy being employed by your immune system to attempt to clear infection. Okay, diphtheria as well. This is another bacterial infection. I'm mentioning diphtheria because it's caused by Corynebacterium diphtheriae, which produces diphtheria toxin. Again, there's this concept of bacteria producing toxins, and these toxins specifically causing disease. In diphtheria, it's essentially an infection of the pharynx, where you get a accumulation of this thick layer of dead cells and some other things as well, but it's essentially observed as a grey mass at the back of the throat, and it's directly caused by this diphtheria toxin, which is released by diphtheria. Okay, so next up I want to talk about viruses. So these are the next group of microorganism that we're going to discuss. Very different to bacteria. The first obvious difference is that viruses are not cellular. So I was talking about the differences between eukaryotic cells and prokaryotic cells. Viruses are completely outside this system. So they're not composed of eukaryotic cells, they're not composed of prokaryotic cells. They're acellular, they're not composed of any cells at all. All viruses is a genome, so just a very small genome enclosed within a protein shell, and sometimes this protein shell is enclosed within a lipid envelope, and that's it. That's essentially all they are. There's some other little accessory bits and bobs. There's some protein sticking on the outside sometimes, but fundamentally that's all they are. Genome wrapped by a protein shell. Sometimes that's it. Sometimes there's a lipid envelope on the outside. How can microorganisms that are so simple, that don't have any means of energy production, that don't have any means to replicate themselves, how can they survive? Don't have any means to transcribe or translate their own genome. How do they exist? It's an interesting question, a big question, and one that we're going to answer in this module. So this is essentially what a virus looks like. This is we call each individual copy of a virus a particle. We can't use the word cell. It's not a cell. It doesn't meet the criteria to be defined as a cell. Some students they'll use the term virus cell in essays, and it's not right. I even read a book once where a person who'd written a book and published it, talked about an Ebola virus cell. It doesn't make sense. It's not correct. It's not accurate. Particle is the correct term. So this here is HIV particle. It's a particle of HIV. And you can see we have the genome in the middle, and this genome is wrapped by a protein shell. We tend to refer to this protein shell as a capsid, and outside of this capsid we have just a bit of stuff, some proteins, some other bits and bobs, but really the next significant structure is this, what we call the lipid envelope. So this is a lipid kind of layer that surrounds the virus particle. HIV has this, a lot of viruses don't. So a lot of viruses will literally just have the genome and then protein wrapped around this genome. I mentioned how simplistic viruses are in terms of not being able to replicate their own genome, not being able to express their own genome. So they can't make proteins, they can't produce energy. The consequence of that is that we consider viruses inert. So when a virus is on its own, when it's not infecting anyone, it's not infecting any cells, it's just on its own, it's inert. It's dead isn't quite the right term, because dead is it's a bit emotive, it's a bit evocative, but inert is the best term. It's not undergoing any metabolic processes. It's not doing any other processes that define life. And that's really important we understand that. The reason it's really important we understand that is because it is why viruses are obligated to infect cells. So the only way a virus can replicate is if it infects a host cell. And that is a consequence of it not being able to do any other life processes itself. As I said, it can't replicate its genome, it can't produce proteins, it can't create energy. However, once it's inside a host cell, it can manipulate the host cell's ability to do that. So it can steal bits and bobs from the host. It can use host cell machinery. So for example, when a virus enters into a host cell, when, for example, an adenovirus that causes a common cold enters into the epithelial cells of your respiratory tract, it will immediately begin using the cellular replication machinery to make copies of its genome. It will immediately begin using the cell's transcription and translation machinery to express its own genome and to produce the proteins it needs to produce. And that's the fundamental basis of viruses' existence. They're different from bacteria. Bacteria can survive on their own. Bacteria can, they survive on their own all the time. They're colonizing the world, basically. There's bacteria everywhere, isn't there? Viruses, there are a lot of viruses. Viruses are kind of everywhere as well, but they're not replicating. They're just waiting to infect a host. They can only replicate when they infect a host. So their life cycle is a lot more jumping from host to host to host. And this is why, because if they're not in a host, they're inert. Okay, so this example here is HIV. My background is HIV, by the way. I think I told you, so I can talk about HIV for days, but I'll just give you an overview. HIV is probably, with the exception of the exception of SARS coronavirus 2, the most well-known isn't the right term. It's the virus that gets the most research attention. And there's specific reasons for that. Almost half the antiviral drugs that have been developed against HIV. That's an example of how much attention HIV gets. There's this really, really big global push to cure HIV. It's difficult though. It's really difficult. HIV infects host immune cells, and that's why it causes AIDS. That's why it causes immunodeficiency, because when it infects these cells, it kills the cells. So when you have HIV for a long time, which anyone who has HIV does have HIV for a long period of time because it's incurable, they get this gradual loss of immune cells unless they're being treated with antivirals. But assuming we're talking about an example where someone isn't being treated with antivirals, they get this gradual loss of immune cells because the replication of HIV is destroying these immune cells. Ultimately, that means you end up with a non-functioning immune system, which defines you as immunodeficient. Your immune system doesn't work anymore. It can't fight off infections. In this state, you're classified as having AIDS. And when an individual has AIDS, they're susceptible to all manner of other infections. And ultimately, an individual with AIDS will acquire just infections that wear them down and eventually cause death because they just don't have a functioning immune system, because HIV has torn through their immune system and just reduced their immune cell numbers to such a low level that they're not capable of fighting off infection. And all this is happening because HIV has evolved to get into those immune cells, because it needs to get into those immune cells to make copies of itself. We have rhinoviruses as well. So when I was talking about the common cold earlier, I used the example of adenoviruses. Rhinoviruses are good examples as well. Most common colds are caused by either adenoviruses or rhinoviruses. Not all, but definitely most. Same principle. These viruses need to get into a host cell and they need to replicate within that host cell. And while they're doing that, they cause disease. The cells that these viruses infect are epithelial cells of the respiratory tract. So rhinoviruses and adenoviruses will enter into the respiratory tract, enter into epithelial cells. Usually you'll get a localized immune response which causes sore throat and things like that. And that's the way they cause disease, really. Not particularly severe disease, but it's still disease. I wanted to make another point there. Yeah, sorry, that's the point. So we see these very different viruses, HIV versus the rhinoviruses. We see they cause very, very different disease. They infect very, very different parts of the body, but the principles are the same. The principles are the virus needs to get into the host cells, whether it's the immune cells for HIV or the respiratory epithelial cells for the rhinoviruses, and they need to replicate in those cells to make copies of themselves. And in doing so, they're causing disease. Same for rotavirus that infects the intestinal epithelial cells. Same for and causes diarrhea. Same for poliovirus that infects the motor neurons and causes paralysis because it's infecting the motor neurons and it's killing the motor neurons as it does it. Same for hepatitis B virus, which infects the hepatic cells of the liver, and in doing so, by various mechanisms, results in the death of these hepatic cells, which eventually leads to liver damage, manifesting as cirrhosis, so liver scarring. That's how hepatitis B causes cirrhosis of the liver. Principles are exactly the same. These viruses have evolved to gain access to the cellular environment, and in doing so, they cause disease. Okay, next up we have fungi. Fungi are eukaryotic. So you remember at the start of the lecture, I talked about the distinctions between prokaryotic and eukaryotic life. You'll also remember that when we're talking about prokaryotic and eukaryotic, we're talking about cellular life. So we're excluding viruses. Fungi are cellular and they are composed of eukaryotic cells. Interestingly, fungi can be single-celled or multi-celled, so unicellular or multicellular. We can compare them to the prokaryotes, so the bacteria and the archaea, and say that prokaryotes are always single-celled. Bacteria and archaea are always single-celled, always unicellular because they're prokaryotes. Fungi, on the other hand, are eukaryotes and they exist as single-cellular microorganisms and multicellular organisms. You may know this already. I'm sure you're aware of multicellular fungi. Mushrooms are multicellular fungi. Single-cellular fungi, the best example is yeast. So the yeasts that we use in food production, these are single-cellular fungi. You might be thinking, okay, so if mushrooms are fungi, why are they microorganisms? Because the criteria for microorganism, micro is small, microorganism means you can't see it with the naked eye. You can see a mushroom with the naked eye. So why is it a microorganism? The answer is some fungi you can't see with the naked eye, the unicellular ones, and even the ones that you can see, they have significant portions of their life cycle that are microscopic, which, you know, that's not unusual for big organisms, but we include fungi in the microorganism group anyway, partly because we have, again, the yeasts that are single-celled, and also just because it kind of makes a bit of sense too, because these fungi cause disease and a lot of other microorganisms cause disease. So even though fungi are in many cases macroscopic, so they're visual with the naked eye, they can be seen with the naked eye, it still makes sense to include them as microorganisms because they cause disease in much the same way as true microorganisms. This is the tree of life that I've been talking about. I don't particularly like this visualization. I'll show you a new visualization next lecture, but some students like this one more than mine. Absolutely fine. It's weird. It just, some visualizations work better with some people and some people don't like those same visualizations. It's just, I don't know what it is, the way, it's just preference, I suppose. It's just preference. So if we go to future lectures and the tree of life that I'm using doesn't seem intuitive, it's just doesn't click. If you come back to this lecture, everything I'm saying will apply to this tree of life as well. It's just a different graphic. You'll see we have bacteria, we have archaea, and we have eukarya. So just like I said earlier, these three domains of life, bacteria and archaea are prokaryotes, eukarya are the eukaryotes. Fungi, as I also said, fit into the eukarya domain. You can see them here next to animals. So they're a separate kingdom to us, which isn't surprising. They're very, very different to us, but they are in the same domain. They're in the eukaryotic domain. So are plants as well, because animals and fungi and plants are all composed of eukaryotic cells. Okay, so we can subdivide fungi up amongst themselves, and the most useful division is filamentous versus yeasts. When we're talking about yeasts, as I said, we're talking about the microscopic unicellular fungi. When you look at them under the microscope, they just look like little circles or little spheres, sometimes diploid, so sometimes you'll see two kind of linked. Very distinct from the filamentous fungi. The filamentous fungi grow as these complex networks of what we call hyphae that branch into a mycelium. So the mycelium is the overall network. This includes the molds. In fact, we sometimes refer to the filamentous fungi as molds. We're also including mushrooms in there, however. It's pretty clear that mushrooms aren't yeasts. They're not single-celled. They're not little round things that you need to view under the microscope. They're actually filamentous. They're actually, even though they look quite ordered and they look quite structured, they are fundamentally a complex network of eukaryotic cells. Next up, we come to the protozoa. Protozoa are a bit of an unusual group because they're a little bit of a catch-all. They're eukaryotes, so all members of the group protozoa are eukaryotic. If you go back to our tree of life, you'll understand what we mean by that. And they're all single-celled. And really, that's about all they have in common in most ways. Basically, if a microorganism is single-celled and it's eukaryotic, we lump it into protozoa. Well, first off, we lump it into a bigger group, which is called protists. But the ones that actually cause disease, we shove them into protozoa. Protists, it's not a term we use very much. It's another catch-all term for all single-celled eukaryotes. Protozoa is a subgrouping of protists, and protozoa, many protozoa are responsible for some quite serious human diseases, and that includes diseases that you may kind of be peripherally aware of, like leishmaniasis and trypanosomiasis and cryptosporidium, but it also includes malaria, which I'm very sure you're aware of. The reason why this unusual group of microorganisms, seemingly tucked away from the others, causes such serious and well-known diseases is interesting. It's very interesting to look particularly at their life cycles. These protozoa have really, really weird life cycles, and these life cycles are inherently linked to how they cause disease. There's very little I can say in a blanket way about protozoa because they're quite different from each other. What we need to do is look at the individual ones that cause serious disease. So that's what we're going to do. We're going to look at plasmodia, which cause malaria, and we're going to look at leishmaniasis and trypanosomiasis and cryptosporidium as well. And we're going to understand the protozoa that cause them. Again, the key takeaway there is what they have in common, which is they are eukaryotes and they are single-celled. Some more examples here, and again I'm just kind of emphasizing the diversity in terms of the regions of the body where protozoan infections can be experienced. So we are going to go into detail about each of these protozoa and and look specifically at how they cause disease. Finally, we have the parasites. Now, parasites is a weird word because we talked about the relationship between host and parasite quite recently, and I introduced the idea that a lot of microorganisms are parasites. A lot of viruses are parasites, for example, because they engage in a parasitic relationship with their host. They cause disease, the host suffers, the virus benefits. That's a parasitic relationship. However, we use the term parasite in a different way as well, because we use the term parasites to specifically refer to multicellular eukaryotic organisms that cause disease. And a lot of these organisms are not microscopic. However, we still talk about them in microbiology, partially because, similar to fungi, they have stages in their life cycle that are microscopic, and partly because they behave like microorganisms in the way that they cause disease. They infect us just like the true microscopic microorganisms do, so it makes sense for us to treat them like microorganisms. What I'm referring to when I'm saying the word parasite is firstly a wide range of parasitic worms. So the cestodes and the trematodes and the nematodes. Again, we'll talk about those distinctions in detail. They're parasitic worms, essentially. They're worms that use us as hosts. Again, they're macroscopic, but they have the stages of the life cycle can be microscopic. Regardless of whether or not they're microscopic or macroscopic, they cause disease in a way that is very similar to the true microorganisms, similar to some arthropods. So hopefully I talk about them on the next slide. Yep, here we have scabies, and here we have head lice and nits, so pediculosis. These are infectious diseases caused by organisms that not really microscopic. I mean, you can see nits and things like that under the naked eye, but it's almost beside the point because they behave in exactly the same way as microorganisms. In the way they cause disease, they find a host, they infect the host, they cause disease, and then they potentially move to a new host. So that's why we have to consider them within microbiology, and that's why we almost have to broaden our idea of what microbiology is. And I know when we think of microbiology, we say it's a study of microscopic organisms, and that's even how I defined microbiology at the start of the module, and that is absolutely true. We just need to broaden our understanding of what we mean when we say microscopic organisms to include some organisms that aren't actually microscopic. I know it's a tiny bit confusing, but hopefully I've justified why we do that. It just makes sense. It makes sense to consider these microorganisms. Sorry, it makes sense to consider these organisms alongside the true microorganisms. Finally, we have something really weird. We have the unusual infectious agents. These are, again, I can't use the term microorganisms because not only are they not microorganisms, they're not organisms. We don't really know what they are. I'll just tell you what they are and you can decide what classification we should give them. The classification I'm giving them and the classification that they are often given is that of unusual infectious agents. That is because they infect, they cause disease like infectious microorganisms. However, then they're not actually microorganisms. They're far too simple. They don't meet the criteria to be defined as viruses or bacteria or fungi. There's something very different. Firstly, we have the prions. Now, prions cause Creutzfeldt-Jakob disease, CJD, which you may be aware of. They also cause things like fatal familial insomnia, and mad cow disease in cows. They're not bacteria, they're not fungi, they're not viruses. All a prion is a single protein. And this single protein is able to infect and spread. When we first found out about prions, it was pretty unbelievable. No one really understood how a single protein could be infecting from person to person. It didn't make sense. Viruses have evolved such complex mechanisms to infect their hosts. Bacteria are way more complex than viruses. They have the ability to do a lot of the things our cells do. Not all of them, but a lot of the things our cells do. So how can a single protein be able to infect host to host? It's a good question, and it's one we're going to explore in the module. Viroids and satellites are similar. These are somewhere between, they're somewhere lower than a virus, so they're subviral. Somehow they lack some of the core features that viruses have, and they're still able to infect. So a viroid, for example, is literally just a piece of RNA. So it seems to infect in the same way a virus does, but it's just a piece of RNA. It's counterintuitive when you understand what viruses are, when you understand how complex the viral life cycle is and how well it has evolved to specifically infect hosts, and how efficient this evolution is and how efficient its infection is. It's crazy to think that viroids, which are literally just a little strand of RNA, can infect host cells. But we're going to explore how and why that happens, because it definitely does happen. Satellites as well. Satellites are somewhere between a viroid and a virus. We'll explore them. The reason why we need to explore these is because prions cause a significant amount of serious human disease. Any prion infection is 100% fatal. Obviously extremely important. Satellites, they're implicated in some human disease. They're implicated in hepatic disease. So satellites can make existing hepatic infections worse. Oh, sorry, just knocked my mic. They can make some existing hepatic infections significantly worse. Viroids, they only really infect plants, but they can infect crop plants, so they can have significant economic impact. It's kind of easy for us to say, oh, it only infects potatoes and tomatoes, who cares? But if you're a farmer and your entire livelihood is dependent upon potatoes and tomatoes, then you care a lot, because you're potentially going to lose your entire income if a viroid spreads. Potato spindle tuber viroid is the name of the one that infects potatoes. But we'll talk about that in a lot more detail. Okay, so hopefully this has been useful. Again, it's just an introduction to the key concepts, but we'll go through each of these specific groups of microorganisms in a lot more detail as the module progresses. Okay, thank you very much.